All right, welcome back, everyone. Last little bit I have to review for the cell division test is how cells are actually regulated when they divide. Um, Cells do need a regulation mechanism. We know what can happen if cells are not told when to regulate. They're when they divide or go through mitosis. If a cell divides too much or is not told when to divide or when to stop dividing, then you can end up with cancer cells. So what happened is in the cell's life cycle, over time there have been built-in what we call checkpoints. So you can see there, a checkpoint is a critical control point where the cell cycle could stop or continue, almost like a red light, green light type of thing. Animal cells will have built-in stop signals, and they can be overridden by the go-ahead signals. You have three major checkpoints. There's the G1, the gap 1 or growth 1 phase, the G2, and M phases for mitosis. The G1 is what we call the restriction point. That's the technical term. I don't get too caught up in knowing the names of the checkpoints. This one, the main idea is that the cell is large enough to divide and that there are enough nutrients available. See, cancer cells really don't care. They don't care about any of this, whether the cell is large enough, whether there's not enough nutrients. Just doesn't care. If the cell does not receive a go-ahead signal in this one, it's going to go into that non-dividing state or the resting state called the G0. Most of our cells are in G0, meaning they don't really have a need to divide unless they're damaged. So, for instance, your liver cells are in G0, but if someone is a recovering alcoholic, they've quit, their liver cells will actually jump back into cell division in the life cycle and start to repair themselves. That is possible. The liver can regenerate. Other fast-growing cells include skin cells, so they will not necessarily go into G0, but most of them do. G2, checkpoint growth 2, that's just to make sure, that's almost our spell check mechanism. So once DNA has been created in the S phase, that has been duplicated, we want to make sure that that DNA is good to go, and we're good to actually duplicate and pass on that DNA to the new cell. So how does this process work? Well, I told you in class, really we separated it into two types of regulation controls. We have internal and we have external. Excuse my typing. Internal control is going, the example I would use would be MPF, mitosis promotion factor, or some call it maturation promotion factor. This is the one that's going to be released inside of the cell to tell the cell, okay, it's all right to divide. It actually all revolves around really two molecules. So if you want to know everything there is to know about internal, right there, internal ways that the body controls when the cell divides and when it does not, it all revolves around these two molecules. You have a CDKs, we call them, or cyclin-dependent kinases. That's an enzyme. How do we know it's an enzyme? Well, we know because it ends in ACE. All enzymes end in ACE. I was waiting for a response there if you didn't catch that. The substrate, or the molecule that binds to the enzyme, remember we talked about a lock and key model? Enzymes only work if the proper key is inserted into the enzyme being the lock. So the proper key for this lock is a cyclin. This is what's actually going to fluctuate and determine whether or not it will bump into the next step and give it the cell will give it the green light to divide. A kinase, just for a little background knowledge, is just a protein that either activates or deactivates another protein by adding a phosphate to them. That's what all kinases do. So how do MPFs actually work? Those are the molecules that actually tell it to divide. Um, Cyclins will accumulate up. Those are the substrates or the keys during G1 and G2. By the time you get to the G2, enough cyclin is available to form the MPF complexes. And I'll show you those complexes in a second on a picture. This is a great one to look at pictures for, by the way. This is going to initiate mitosis. Later in mitosis, the MPF switches itself off by initiating a process that will eventually destroy the key. CDK, the non-cyclin part of MPF, persists in the cell as an inactive form until it associates with new keys or cyclins synthesized during interphase of the next round. Let me show you a picture real quick, because it's a time when a picture speaks 1,000 words. Let me move this over. Okay, so obviously I just Google image this. You can look at this. This is one I have in my PowerPoint. Give credit to wikispaces.pennstateuniversity.edu. Here's your cell cycle G1S, G2. Notice on your graph to the left you have concentration on the Y. Here's your, on the X you have the different stages of cell cycle. Notice the peak of the MPF, that's the mitosis promotion factor. It peaks right before mitosis, and that's what gives us the go-ahead for mitosis. But look what activates this. So these cyclins, those were the keys that fit into the enzyme, the substrate. As cyclin increases throughout the life of the cell, you have more keys binding to the actual enzyme or the lock. And when that happens, it phosphorylates the next protein, which bumps it to the next checkpoint, which eventually, at the very end, you have a fully active MCDK complex. It's going to release MPF and kickstart the cell into mitosis. So that right there, all in all, is how the cell regulates internally when it divides. Those are the red light, green light. The only other type I offer up as an example of external, I'm going to go back here to condensed notes. Here's your external, so let me label that here. This is your external regulatory factor. It's called PDGF, or platelet-derived growth factor. So this one, the biggest thing, it's the way that we call it external is because it's usually used for wound healing. So if, let's say you get a cut, when you have an injury, your body is actually going to release this factor. These little things, fibroblasts are connective tissue which possess PDGF receptors on their plasma membranes. So when this PDGF is released, when it binds to these special cells, it's going to lead to a massive creation or a kickstart or proliferation of these fibroblasts, and that's what's actually going to help you clot and heal the wound. So that's an external thing. Yeah, I mean you could be injured multiple ways. There can be a burn, both heat, temperature, chemical burn. You could be cut. So that's a way that you could have an external stimulus causing a regulation of when that process will be turned on. Just as a little aside as well, density-dependent inhibition may be something you want to look at. Sometimes that can be mentioned on the AP exam. It's basically just talking about how typically cells, a healthy cell will grow. Let's say you're growing E. coli in a petri dish. It'll keep growing and growing and growing until literally that whole surface of that petri dish is covered with E. coli. When there's no space left, they will actually stop. The density is too high, and at risk of killing their partners, I guess you could say, and using up all of the space and nutrients, they'll stop dividing. That's density-dependent inhibition. They're inhibiting their growth due to how many of them there are surrounding them. They don't want to use up all the nutrients. And of course, you have cancer cells when they do not exhibit any of these control factors. And you can look a little bit more into the notes on that. Other than that, that wraps up all of mitosis meiosis. Check out the PowerPoints and rewind this if you have to. Check out your condensed notes, and I think you should be pretty well prepared for the cell division test. Thanks.